We are about to start today's session, that is biological oxidation and electron transport chain. Okay, that is electron transport chain. So this electron transport chain, here the electron flow occur across the successive dehydrogenase enzyme, which is known as the electron transport chain. Here the electrons flow from the electronegative potential to the electropositive potential. So here the highest electropositive potential is of oxygen, that has a capacity to accept the electron. So oxygen that we inhale in our respiratory system, that will carried by the in the lung, and then with the help of hemoglobin it reach to the different tissue, where finally this oxygen will accept the electron and it will help us to produce the ATP. That's why oxygen is very, very important. Otherwise, if we don't inhale oxygen in our breath, then our mitochondria, peripheral tissue mitochondria will not produce ATP. Without ATP, you know that muscle contraction is not possible, nervous system is sodium potassium pump is not possible, or many synthetic pathway is not possible. So the cell will die in absence of ATP. That's why this topic is very, very important. While electronegative potential, that is of NADH, that is the reducing equivalent. What is the which vitamin? NADH is a form of which B complex vitamin? NADH is the active form of which vitamin? Whenever you listen this word NADH, that will be active form of which vitamin? Yes, excellent. Vitamin B3, right? So electronegative potential is highest, that is minus 0.32. That means it will donate the electron. Okay, so that electron flow from NADH to the oxygen through the electron transport chain. This electron transport chain is present at the inner membrane of the mitochondria. Remember this. So if I draw the bigger picture of the mitochondria, okay, this is a, what is this? This of mitochondria, this is the outer mitochondrial membrane, right? Then inner mitochondrial membrane will be in the form of crystal. Okay, this is inner mitochondrial membrane. This is, what will be this? Write in the chat box. This is called as, no voice? Okay, yes, it is matrix. Okay, and this is intermembranous space. All right. So electron transport chain is located at the inner mitochondrial membrane. So here it is in the form of 4 complex. So suppose this is complex 1. It is made up of 1 proteins, enzymes. So complex 2, complex 3, and complex 4. Okay. The first complex is called as the NADH coenzyme Q complex. Okay. Second complex, it is inbuilt inside the inner mitochondrial membrane. It is FAD coenzyme Q complex. This complex is called as a cytochrome C complex, and here this is cytochrome oxidase complex. Okay, this is little complex, you will learn. So here there are 2 carriers. One is coenzyme 2 mobile carriers. Okay. So what will be the role of these mobile carriers? It will take coenzyme Q, which is also called as a ubiquinone. Coenzyme Q is called as a ubiquinone. It will take up the electron from the complex 1, okay, and it will transport the electron to the complex 3. All right, remember this. While this complex coenzyme Q will also carry the electron from the complex 2 also, okay, and from complex 3 to complex 4, there is another mobile carrier that is called as a cytochrome C. Okay. So if I draw the diagram again, This is outer mitochondrial membrane. This is the inner mitochondrial membrane. Okay. So here there are 4 complex: complex 1, complex 2, complex 3, complex 4. Now, whatever diet that we take, either in the form of carbohydrates, fats, and proteins. Okay, carbohydrate, fat, proteins. Then this will be ultimately, as we know, carbohydrate will go to the glycolysis and ultimately produce the pyruvate. Pyruvate will be converted to acetyl-CoA. Okay, and acetyl-CoA go to the TCA cycle. Okay, and in between, while conversion of glucose to the pyruvate, it will produce 2-NADH, right? Also, when TCA cycle, it produces 3-NADH. Also, we will learn in fatty acid, when fatty acid is oxidized, it will also produce NADH. From one palmitic acid, around 7 NADH are produced. So this NADH, they will carry the electron. How? So now you have to understand one concept called oxidation and reduction. So what is oxidation? Anyone can write? What is oxidation? What is oxidation? What is oxidation? Yes, very good. It is loss of electron. Okay, it is also called as a LEO. Okay, you can remember it is LEO. So oxidation is loss of electrons or hydrogens. So whenever there is a loss of electrons or hydrogen, it is called as the oxidation. And what is reduction? What is reduction? Yes, excellent. It is gain of electrons. It is gain of electron or hydrogen. Okay. So, like in glucose, if you see the structure of glucose, there is a one side there is a OH group, one side there is a H group. So here hydrogen atoms are removed, so it is called as a oxidation. So we see that it is an oxidation process. Okay, loss of hydrogen. Okay, and this hydrogen are carried by the NAD. Okay, so here glucose, they will donate its 2 hydrogen atoms and NAD will gain the hydrogen. So one will be oxidation and here there is a reduction. So NAD will gain 2 hydrogen atoms. So if we write it for the hydrogen atoms, you can say 2H plus and 2 electrons. Understand? If you want to write hydrogen, you can write 2 protons and 2 electrons. Then you can write it like H plus plus H plus plus 2 electron minus. And this H plus and 1 proton and 2 electron, you can write it like H minus, that is hydride ion. And NAD plus and H minus, they will form the NADH plus H plus. So if we see when NAD it is reduced, that means it will gain the hydrogen atoms or gain the electrons. Hydrogen atoms will contain the electrons. So when it gains the electron, NAD is reduced to NADH plus H plus. So you will see everywhere in biochemical reaction that NAD, it is converted to NADH. So that process is called as the reduction. I hope this concept is clear. While glucose, they will donate the hydrogen ion. So it is called as the oxidation. Glucose is oxidized while NAD is reduced. So NADH is in the form of reduced. It has so much amount of electron. You can see here it contains the electron. NADH is in the form of electron. So it is a donkey, like it is like donkey which carry the electron. NADH is a donkey that will carry the electron. Okay. So NADH, where we get the NADH? From TCA cycle or fatty acid oxidation or even proteins, amino acids, they are broken down. Some amino acids, they are broken down to acetyl-CoA. So acetyl-CoA is the ultimate, okay, molecules, and when it go to the TCA cycle, it produces the NADH again. So this NADH, they will transfer their electron to the first complex, which is made up of FMN and FES. Okay, what is FES? Is the iron sulfur complex, and FMN is the active one of the complex part of the vitamin B2. So NADH give its electron to the first to the FMN, then to the FES. We will see, don't worry, and then finally this FES will donate its electron to the coenzyme Q. So first from the NADH, electrons are transferred to the FMN, then to the FES, and then FES will transfer its electron to the coenzyme Q. Then what will happen? This is very important short note, little bit complex, students. Please pay attention. It is a most complex topic. Okay, I will try to make it simple, but as such it is very complex. Okay, don't worry, but we will try to make it easy. Then this coenzyme Q will transport its electron to the complex 3. Okay, and here there is a cytochrome B. It will transfer its electron to the cytochrome B, then to the cytochrome C1. Okay, and then this cytochrome C1 will transport its electron to the another mobile carrier. One mobile carrier is cytochrome coenzyme Q, another is cytochrome C. Okay, so coenzyme Q to the cytochrome B, C1, and then cytochrome C. Then this cytochrome C, it is a mobile carrier. What it will do? It will take up the electron from this C1 and it will transfer it to the complex 4. Okay, and then it will transfer its electron to the cytochrome A and AA3, and ultimately it will transfer its electron to the oxygen. So you can see there is the potential, electropotential for NADH is in negative. It is like 0.32. So it will donate the electron. Okay, and subsequently the electropotential of this different compound, so that they can transport its electron from one component to the another. While oxygen has the highest electropotential, that is 0.82, so it will accept the electron maximally. So it is the ultimate electron lover and it will convert to the H2O. Okay. So here electrons from the reduced coenzyme like NADH and FADH. For the complex 2, it will transfer the electron from FAD to FES and then coenzyme Q, and then complex 2 to complex 3 and complex 4. Complex 1 is bypass in case of when electrons are transferred from the FAD. Okay. So it will pass through the electron transport chain before they reduce the oxygen from water. Okay, that is the last step. The passage of electron through the ETC is associated with the loss of energy, which is utilized to generate the ATP. Okay, don't worry, we will see of this. So you can see here NADH, it is like donkey. It contains the electron. It has a lots of electron, NADH, right? It will transfer its electron to the complex 1. That will transfer first to the FMN, then FMN will be transferred to the FES, and then FES will transfer its electron to the coenzyme Q. Okay. Then this coenzyme Q will transfer its electron to the complex 3, that is cytochrome B and C1, and ultimately cytochrome C. Then this cytochrome C will transfer its electron to the cytochrome A, AA3, and ultimately oxygen will accept this electron and hydrogen, and it will form the H2O. Okay. So you can see glucose will convert into pyruvate, fatty acid will be oxidized to acetyl-CoA, amino acid, their carbon skeleton will form the acetyl-CoA. And this acetyl-CoA to the TCA cycle, and they will produce the NADH and FADH. That will go to the electron transport chain, 1, 2, 3, 4 different complex, and ultimately they will form the oxygen will take up the electrons and they will form the H2O. And during this ATP is produced. How this ATP are produced, we will see. Okay. So inner mitochondrial membrane contains 5 separate complex 1, 2, 3, 4. 5 is for the ATP synthase. 5 is ATP synthase. While complex 1 to 4, they are part of electron transport chain. So there are two mobile carriers. Which are they? One is coenzyme Q. One is coenzyme Q. And another is cytochrome C. What is the other name of coenzyme Q? I have already told. Please write down so that I know you are listening. What is the other name of coenzyme Q, which is a mobile carrier? What is the other name of coenzyme Q? Yes, excellent. Excellent. Abhishek, Drumil, Gray. Okay, thank you all of. So other name is ubiquinone. Remember. And you know ubiquinone is synthesized from the farnesyl pyrophosphate, which is synthesized during cholesterol biosynthesis. So when cholesterol is synthesized during intermediate pathway, there is a farnesyl pyrophosphate, which is a precursor of this coenzyme Q. That's why in patient where there is a hypercholesterolemia, we give the drug called statin. Have you heard of this name in competitive inhibition? Enzyme competitive inhibition. Have you heard this name, statin? It is a competitive inhibitor of which enzyme? Statin. Drugs are given like atorvastatin, lovastatin. It is a competitive inhibitor of one enzyme. It inhibits which enzyme? Yes, very good. But not HMG-CoA. What is the name of exact enzyme? HMG-CoA. आगे क्या आएगा उसमें? Statin will inhibit HMG-CoA reductase. HMG-CoA reductase. Not synthase. HMG-CoA reductase. That will convert HMG-CoA to the mevalonate. Okay, and mevalonate will produce FPP and ultimately cholesterol is synthesized. So if we inhibit this enzyme, then cholesterol biosynthesis is reduced. So FPP is also reduced. So ubiquinone will not be synthesized. So that will be the cause of statin-induced myopathy because muscle will not produce ATP when given for the long term. Okay, short term के लिए नहीं लेकिन long term तक देते हैं तो there is a chance of myopathy in because of the statin drug. Remember ये medicine में भी काम लगेगा आगे भी, ठीक है? और general practice में statin may induce myopathy because ATP will not be produced because ubiquinone will not be produced. Okay. So now we are seeing each of the complex. So you can see NADH, that is reduced form. Okay, it will give its electron to NAD and itself it is oxidized. So here there will be NADH to NAD. This process is called oxidation. NADH is converted to NAD. So NAD is in the reduced form and NAD is oxidized form. While FMN is converted to FMNH2. Okay, as we have seen that NADH will give its electron to FMN and FES, and then it give its electron to the coenzyme Q. NADH electrons are transferred from NADH to FMN, then FES, and lastly coenzyme Q. So FMN, what it will be? It will be oxidized and it will be converted to FNH2. So now FMNH2 is carrying the electron. So it will be called as a reduced form. FMNH2 is called reduced form. Now reduced form it donates its electron. So it will donate its electron to the oxidized FES, and oxidized FES will be converted to reduced form. Okay. Or you can see here there is a Fe plus 3 which will be converted to Fe plus 2. So this reduced form has now electron, and it will give its electron to the coenzyme Q, and coenzyme Q will be converted to QH2. Okay, that is reduced form QH2. याद रखो कि जब भी H2 फॉर्म होगा तो वो कहलाएगा H होगा या H2 होगा, that will be reduced form. So ultimately now QH2 देखा आपने, यहाँ पे NADH था, उसके पास इलेक्ट्रॉन था और वो इलेक्ट्रॉन अभी आ गया है QH2 के पास. ठीक है. So now the reduced form of the ubiquinone or Q, coenzyme Q, has the electron. Now it can donate its electron to the another component. Okay, so NADH coenzyme Q reductase complex accept the electron from the NADH and pass the electron to the next electron carrier. तो आपको लिखना ये पूरा कि समरी क्या होगी, इलेक्ट्रॉन फ्लो फ्रॉम द NADH, फिर FMN, FES और Q. Okay, during this process here energy, when there is a transfer of electron, during the flow of electrons, here there is a free energy is released that will drive 4 proton pump out of mitochondria. इसका मतलब क्या हुआ, suppose this is a mitochondria, this is the inner mitochondrial membrane. So during, suppose यहाँ पे electron transport chain है, so during the flow of electron from complex 1 to the coenzyme Q, 4 protons are pumped from the matrix to the intermembranous space. तो यहाँ पे 4 प्रोटॉन पंप करेंगे because of energy that is released during the flow of electron. That you have to remember. Now this complex 2 we will see in some time. Okay, now coenzyme Q, which is reduced form, it has now electron, so it will donate its electron first to the Fe+3, that is oxidized form, and itself it will be converted to FES, that is Fe+2 form. Now Fe+2 form is now reduced, so it will donate its electron. So it will donate its electron to the cytochrome B. Cytochromes are nothing, that is it contains the iron. Okay, so cytochrome B, that is oxidized, it will be converted to reduced form of the cytochrome B. Now cytochrome B has an electron, it will give its electron to the cytochrome C1, and cytochrome C1 itself will be reduced. Okay, and ultimately another mobile carrier, that is cytochrome C, it will take up the electron and itself it is converted to reduced form of cytochrome C. So यहाँ पे क्या याद रखना है, Q से आगे FES, फिर cytochrome B, cytochrome C1, और अल्टीमेटली जो मोबाइल कैरियर था cytochrome C, that will take up the electron. Okay, are you getting? क्या हो रहा है यहाँ पे, देखो ये माइटोकॉन्ड्रिया है, ये इनर माइटोकॉन्ड्रियल मेम्ब्रेन, दिस इज इनर माइटोकॉन्ड्रियल मेम्ब्रेन, this is complex 1, suppose this is complex 3, this is complex 4. So NADH, that is produced in mitochondria matrix. Okay, during TCA cycle, where TCA cycle takes place, where fatty acid oxidation takes place. Okay, fatty acid oxidation, we will learn that fatty acid oxidation takes place in the mitochondrial matrix. TCA cycle also takes place in the mitochondrial matrix. So whatever NADH or FADH2, okay, FADH2 will go to the complex 2. So NADH that is produced during TCA cycle or fatty acid oxidation, it will give its electron. Okay, so यहाँ पे देखो आपने गॉड ने कितना जबरदस्त बनाया है, यहाँ पे ऑलमाइटी गॉड ने क्या रखा है कि NADH मैट्रिक्स में एंजाइम होते हैं, वहां पे NADH बना, वो तुरंत ही देखा आपने इनर माइटोकॉन्ड्रियल मेम्ब्रेन पे ही इलेक्ट्रॉन ट्रांसपोर्ट चेन रखी और कहीं पे नहीं रखा है, वहां उसको फिर ट्रांसपोर्ट होना पड़ता, साइटोप्लाज्म में जाना पड़ता. सपोज ये पूरा हमारा सेल है बॉडी का, ठीक है, तो आप समझो कि अगर लाइसोसोम में ये होता तो क्या होता, कितना प्रॉब्लम होता. तो क्या होता है, NADH इट गिव इट्स इलेक्ट्रॉन टू द कॉम्प्लेक्स 1. कॉम्प्लेक्स 1 में क्या होगा, पहले क्या आएगा, FMN इलेक्ट्रॉन लेगा, इटसेल्फ विल बी कन्वर्टेड टू रिड्यूस फॉर्म, फिर उसका जो रिड्यूस फॉर्म होगा वो डोनेट करेगा इलेक्ट्रॉन किसको, FES को, और फिर क्या करेगा, यहाँ पे कोएंजाइम Q होता है मोबाइल कैरियर, इट विल टेक अप द इलेक्ट्रॉन. ओके, और वो क्या करेगा, डायरेक्ट दे देगा कॉम्प्लेक्स 3 को. ठीक है, अभी कॉम्प्लेक्स 3 में क्या आएगा, सबसे पहले FES ऑक्सीडाइज होगा, वो रिड्यूस होगा इलेक्ट्रॉन लेके, फिर साइटोक्रोम B, फिर साइटोक्रोम C1. ठीक है, नाउ साइटोक्रोम C1 गेट्स इट्स इलेक्ट्रॉन. अभी यहाँ पे कौन इलेक्ट्रॉन्स लेगा साइटोक्रोम C1 से, साइटोक्रोम C1 डोनेट्स इट्स इलेक्ट्रॉन टू विच मोबाइल कैरियर, राइट डाउन. दूसरा कौन सा मोबाइल कैरियर है, वेरी गुड, साइटोक्रोम C. ठीक है, साइटोक्रोम C विल गेट्स इट्स इलेक्ट्रॉन फ्रॉम C1 एंड वो किसको डोनेट करेगा, कॉम्प्लेक्स 4 को, जहां पे साइटोक्रोम A3 है. इट इज साइटोक्रोम A3 इज द कॉपर कंटेनिंग कॉम्प्लेक्स. ठीक है, और अल्टीमेटली A3 से कहा जाएगा ऑक्सीजन, इट विल गेट द इलेक्ट्रॉन विद द अल्टीमेट इलेक्ट्रॉन एक्सेप्टर. ठीक है, इट इज अ वेरी गुड इलेक्ट्रॉन लवर एंड इट विल कन्वर्ट इट टू द H2O. So you can see here cytochrome C, that is a mobile carrier. It will get the donates its electron itself converted to oxidized form, and it give its electron to the cytochrome AA3 complex. Okay, आप देख सकते हो that is oxidized form which itself is reduced to cytochrome, reduced form of cytochrome AA3. Now this reduced form, so it will donate this electron, and oxygen is the ultimate electron acceptor because it is the electropotential of 0.82. It will take up the electron with the help of H plus, and it will form the H2O. It is a water. Okay, understand? So cytochrome C to AA3 and oxygen. What will happen about the complex 2? So three major enzyme systems that transfer their electron directly to the ubiquinone from FAD prosthetic group. Okay, so one is succinate dehydrogenase, one is FAD. This is enzyme is for the, आप पढ़ोगे fatty acid oxidation में आएगा पहला ही step. ठीक है, succinate dehydrogenase is also a part of TCA cycle. So succinate to fumarate, FAD is converted to FADH2. So from succinate, it will donate its electron to FAD. FAD itself is converted to FADH2, that is a reduced form. Now this FADH2 will carry the electron, so it will convert oxidized FeS to reduce Fe plus 2. Okay, so Fe3 plus में से यहाँ पे Fe plus 3, that is converted to Fe plus 2, and it give its electron to the coenzyme Q, and coenzyme Q itself will be reduced to QH2, that is a reduced form of coenzyme Q. फिर क्या होगा? Coenzyme Q will go to the complex 3 and complex 4. ठीक है? So you can see in the diagram when from pyruvate to acetyl-CoA, alpha-ketoglutarate to acetyl-CoA, okay, isocitrate to alpha-ketoglutarate, malate to oxaloacetate. Here NADH is produced. Okay, that NADH will be converted to NAD, and it give its electron to complex 1, that will be FMN and FES. Sorry. So during this process, during the flow of electron, energy is released, which will drive the 4 protons out. Remember this. Okay. फिर coenzyme Q, it will give its electron to the complex 3, that is FES, cytochrome B, cytochrome C1, and then ultimately it will transfer electron to the another mobile carrier, that is cytochrome C. During this flow of electron, again energy is released. This energy is sufficient to drive 4 protons pump out. So again 4 protons will be pumped out from, what will be this? This is a matrix. This is the inner membrane. This is the intermembrane space. So from matrix, 4 proton again will pump out. Then cytochrome complex 4 to the oxygen. Again the energy is released. That energy will drive, it is not that much, so it will drive 2 protons pump out. So you can see total 4, 10 H plus 10 protons pump out from matrix to the intermembranous space during the flow of electron from NADH. While what will happen to the FAD? FAD, there is some special thing. FAD is already, this complex, succinate dehydrogenase complex, is already inbuilt in the inner mitochondrial membrane. So here the flow of electron occurs. Like in NADH, there is a complex 1, flow of electron from FES to coenzyme Q occur. Here also the flow of electrons occur, but energy that is released, energy is also released, but that is not sufficient to pump the proton. So from FAD complex 2 to complex 3, there is no proton will pump out. Okay. So no 0 protons are pumped out because energy is not sufficient to pump out the protons. Remember this. So अगर FAD complex 2 से इलेक्ट्रॉन ट्रांसपोर्ट हो गए तो कितने प्रोटॉन पंप आउट हो गए टोटल? From NADH 10 H protons are pumped out. From FAD how much protons are pumped out? Please write down. Excellent, very good. Saurabh, Gray, Keval. Yes. So from FAD, very good. All students are correct. So from FAD, 6H plus, 4 from complex 3 to cytochrome C, and complex 4 to oxygen 2. So total 6 protons are pumped out. So I hope this is clear to you. Now, you can see I am again drawing the mitochondria. Now I am not drawing the cristae. So this is matrix. This is intermembranous space. All right. So this will be complex 1. This is complex 2, complex 3, and complex 4. From the complex 1, 4H plus pump out. From complex 3, again 4H plus pump out. And complex 4, 2H plus are pumped out. Okay. Now here, this proton pumps, okay, they will form the positive gradient. This positive gradient will help in formation of ATP. How? So there is another complex that is called as the ATP synthase. Okay. This ATP synthase, during the flow of protons from inner mitochondrial intermembranous space to the matrix, when H plus go from intermembranous space again to the matrix, it will help to produce ATP. Okay, that is complex 5. Now this is very important. That is called as oxidative phosphorylation. That means coupling of oxidation. What will be the oxidation? During the electron transport chain, that electrons flow from complex 1 to 3 to 4, complex 1 to 4, whatever oxidation reaction takes place, that is coupled with the phosphorylation. That means formation of ATP. So Peter Michel in 1961 first proposed this oxidative phosphorylation. तो अगर आपको एग्जाम में सिर्फ इलेक्ट्रॉन ट्रांसपोर्ट चेन पूछे या बायोलॉजिकल ऑक्सीडेशन अगर आपको पूछते हैं तो आप कॉम्प्लेक्स 1 to 4 तक का आप मेंशन कर सकते हो, लेकिन ऑक्सीडेटिव फॉस्फोरिलेशन अगर एग्जाम में आता है, दैट मींस यू हैव टू मेंशन द कॉम्प्लेक्स 5 आल्सो, दैट वी विल सी नाउ. Okay. So transport of proton from inside to the outside inner mitochondrial membrane is accompanied by the generation of proton gradient. ठीक है. So protons जो इलेक्ट्रॉन ट्रांसपोर्ट चेन में मैट्रिक्स से आउटर intermembranous space तक गए, that will form the proton gradient. This proton gradient will create the electrochemical potential that will drive the synthesis of ATP. Okay. So if we see the diagram of the ATP synthase, okay, I will share the video of ATP synthase in our group. It will be in animated form. एक दो वीडियो है, that will be very easy to understand. ठीक है, थियोरेटिकल मैं नहीं एक्सप्लेन कर सकता हूं, अगर टाइम रहेगा तो मैं वीडियो यहाँ पे भी आपको दिखाऊंगा, वरना मैं ग्रुप में शेयर कर दूंगा तो आपको क्लियर समझ में आ जाएगा. आपको इतना याद रखना है कि सपोज दिस इज द माइटोकॉन्ड्रिया, दिस इज द इनर माइटोकॉन्ड्रियल मेम्ब्रेन. ठीक है, दिस इज मैट्रिक्स, मैट्रिक्स है यहाँ पे. सो कॉम्प्लेक्स 5 इट इज मेड अप ऑफ 2 यूनिट्स. One is FO unit, one is FO unit which is embedded in a FO. Why it is O? It is not F0, it is FO. O stands for the oligomycin, because this FO complex of the ATP synthase is inhibited by the antibiotic called oligomycin. This is also asked in the next exam or NEET PG exam. Okay, oligomycin will inhibit ATP synthase by inhibiting the FO complex. ठीक है, तो ये FO complex होगा which is made up of 10 to 12 peptides. That is peptides of, इसका नाम कहा है C units. It is made up of C polypeptide units. फिर एक आपका आएगा यहाँ पे एक साफ्ट होगा, और दूसरा जो बनेगा कॉम्प्लेक्स उसको कहेंगे F1 कॉम्प्लेक्स. ठीक है, F1 कॉम्प्लेक्स will protrude towards the matrix, while FO complex will be embedded in the inner mitochondrial membrane. तो ये आपका membrane है, this is your matrix. Okay, so flow of proton from this is intermembranous space. From intermembranous space, proton will pump back again inside the matrix, and when protons are go inside this matrix, it will drive the it will get the force, and that will help in the formation of ATP. Okay, we will see. So here this FO unit is made up of proton channel, nothing but a proton, nothing but a protein amino acid that is made up of C units. Okay, and H plus will flow through this. Okay, it is little bit complex. I will share the diagram, that will be clear to you. Then F1 subunits, that you have to remember, it is present inside this matrix, projects into the mitochondrial matrix. It is made up of 3 beta and 3 alpha chain. Alpha chain have a binding sites for the ATP and ADP, while beta chain will help in the catalytical action. Okay, so that is 3 alpha and 3 beta that you have to remember. There is a binding chain mechanism is there. So there is 3 form is there. One is O, T and L. In the L form, there is ADP and PI is there. Okay, but they are loosely attached. When protons flow through this ATP synthase, then ADP and PI, they are bind together and they will form the ATP. So ATP is now formed, and then again this shaft is rotate, and it is open form that will be go out. Okay, so you can see from complex 1, 4 protons are pump out. From complex 3, 4 protons pump out, and complex 4, 2 protons pump out. That protons will go again from intermembrane space to the inside of matrix, and during that process, ADP is converted to ATP. Okay, and this ATP will be carried by the ATP transporter, and whenever ATP is required, it will go. Right? So this is a very important process. So from NADH, 4H plus, so 4H plus protons, when they go inside, they will produce 1 ATP. That's why earlier what was the thoughts that there are NADH electrons flow from the 1, 2 and 3 complex, so 3 ATP are produced from NADH earlier. And from FAD, there is 2 times flow of electrons occur, so 2 ATP are produced. But now researchers have found that exactly 4H plus are required to form the 1 ATP. Okay, so from NADH, how many ATP are produced? Instead of 3, it is 2.5, because 4 from the 1, okay, from complex 3, another 1 ATP, and from 2 protons, only 0.5 ATP. So that's why 2.5 ATP from NADH. And how much ATP will be produced from the 1 molecule of FAD? From FADH, yeah, ATP, how many ATP are produced from 1 molecule of FAD? Yes, very good, Abhishek, Keval, Saurabh. Okay, very good. So from FAD, 1.5 ATP are produced. So I hope it is clear. Earlier, you can say it is 3 ATP from NADH and 2 from the FAD, but now exact researchers have found that 2.5 from the NADH and 1.5 ATP. एग्जाम में आप कोई भी आप लिख सकते हो, लेकिन आपको मेंशन करना होगा कि आपको ओल्ड थ्योरी आप मेंशन कर रहे हो या न्यू. ठीक है? This is what is PO ratio, number of ATP generated from each oxygen that is consumed. It is 3 for the electron from the NADH, 2 for the FADH, or you can see now 2.5 or 1.5. Okay. So 1 NADH will produce 2.5 and 1 FAD will produce 1.5 ATP. Okay. So this is, I think, clear. Now coming to the which are the inhibitor. This is very important for the All India exam. Next exam, कोई भी competitive exam है, inhibitors, they are very important. So this inhibitor will inhibit oxygen consumption and secondary hold the synthesis of ATP. Some of these are poison, some of these are drug. Okay, we will see. So inhibitor of complex 1 is barbiturate, which is used as a sedative or even hypnotics. So barbiturate will inhibit complex 1. NADH coenzyme Q complex को inhibit करेगा. Rotenone Okay, it is an insecticide or it can act as a fish poison. Okay, and pyricidine is an antibiotic. So barbiturate, rotenone, and pyricidine, they are inhibitor of complex 1. Clear? Complex 2 inhibitor, one is carboxyne, and another will be, which is a competitive inhibitor of succinate dehydrogenase enzyme. One is carboxyne, and which is the another. Anyone, please write. Which is another inhibitor of complex 2, that is the inhibitor of succinate dehydrogenase. Yes, excellent. Sonu, very good. Chintan, malonate, right? So carboxyne and malonate, they are inhibitor of complex 2. Then complex 3 inhibitor is antimycin and British anti-levisite. What is this anti-levisite? Levisite was poison that was used in the World War, and British UK person, they have found remedy against it, levisite, that is called BAL. Okay, so dimercaprol is also called as a BAL. So antimycin and BAL, they are inhibitor of complex 3. And complex 4, very, very important, cyanide, carbon monoxide, and azide. These are frequently asked in the next exam or any NEET-PG exam. That which is the inhibitor of complex 4. Okay, this is not 6, this is complex 4. Okay, complex 4 is inhibit mainly cyanide, carbon monoxide. So there is a cyanide poison. You have seen in many terrorists, they are taking the cyanide. They are taking something when they are caught, any terrorist or any antisocial person, when they are caught by the police, they take the drug inside the tongue. That will be mostly a cyanide. So when they take the cyanide, they will inhibit the complex 4. By that, from oxygen, ATP will not be, oxygen is not converted to H2O. So ATP is not produced and person will die within a minute. So you have to take very good care from when you are dealing with the cyanide or anything. So cyanide, carbon monoxide, azide, they are inhibitor of complex 4. Okay, so you can see here there is a complex 1 where NADH will give its electron to the ubiquinone. So this step is inhibited by rotenone, barbiturate, and pyricidine. Remember, from complex here, if there is a complex 2, from succinate to the ubiquinone to the complex 3, this complex 2 inhibited by the carboxyne, and another is malonate. From complex 3 to the cytochrome C, what are the inhibitor? Antimycin and British anti-levisite. Okay, and complex 4 inhibitors, very important, cyanide, azide, and carbon monoxide. Complex 4. Okay, cyanide, azide, and carbon monoxide. So remember this. While ATP synthase, complex 5 inhibitor is oligomycin, while atractoside will inhibit the transporter, ADP, ATP transporter. Once ADP is formed, that will be transported from outside to inside ADP, and ATP will be transported from matrix to the intermembranous space. That is by the ADP transport that is inhibited by the atractyloside. Now you have to remember one last terminology, that is uncoupler. So what is uncoupler? So there are a protein that will leak of the protons. So if we know this is the outer, okay, this is matrix, this is inner membrane. So protons are pumped during the electron transport chain. These are the protons, but due to this uncoupler, there is a channel that will leak the H plus. So that will not produce the proton gradient. So what are these uncoupler? Like thermogenin in round adipose tissue, thyroxine, bilirubin, and essential fatty acid. While synthetic uncoupler are 2,4-dinitrophenol, 2,4-dinitrocresol, and CCCP, chlorocarbonyl, cyanide, phenyl hydrazole. So what is the use of this physiological uncoupler? So they will form the oxidation, but they will not allow the phosphorylation. Uncoupler will not allow phosphorylation because whatever protons are formed in the inner membranous space, they will again go back inside the matrix. So they will not allow the proton gradient. Out of this, most important is thermogenin, that is present in the brown adipose tissue and also in the neonates. So they will produce the heat and they will protect the children from the neonatal hypothermia. So they will produce the heat. So during when animal come from hibernation to outside, they will allow formation of so much energy is required, so much heat is required. So that is very important. So brown adipose tissue is very important. in hibernating animals and also in the neonates. Other physiological uncoupler are thyroxine, bilirubin, and essential fatty acids. Okay. And there are some diseases that are associated with the mitochondria, and you should remember mitochondria, they are inherited, transmitted maternally. Okay, paternal because the upper outer layer of the sperm is taken inside, so they will not give parent, sperm will not give its donate its mitochondria. Only the female cytoplasmic mitochondria that is inherited. So mitochondrial disorder only inherited maternally. So from mother to child it is transmitted, not from father to child. And very important out of this is mitochondrial encephalopathy, lactic acidosis, stroke-like episode, MELAS. Okay, in all this, due to the defective mitochondrial DNA, there is a defective ATP production. And it will lead to the labor, hereditary optic neuropathy, myoclonic epilepsy, and labor. So these three are very important. Then lethal infantile mitochondrial obstruction can be also. So this is all about the oxidative phosphorylation. Okay, I will share further instruction in our WhatsApp group. I will share a video. I will share the PPT also, and also a video of our YouTube channel. I hope it is helpful to you to understand this topic. And once you read through the books, then you will understand. Okay, thank you, students, for your great participation. Thank you. Bye.